Hi, this is Mr. Wartsky, and today we are going to start our adventure into the excretory system in this first screencast of the body systems. Now, we are going to look at the body systems that maintain homeostasis, which is a balance. And hopefully, we'll do some comparative anatomy so you know how it changed over time to be what it is today in Hugh people. So in order to introduce the excretory system, we're going to talk about the two problems that animals had to solve when they moved on land. So cells cannot survive a net gain or loss of water. We need to maintain water concentrations inside of our cells, and we need to get rid of nitrogenous waste. And those nitrogenous wastes come from amino acids. So we're going to talk about two things when we talk about the excretory system. One is osmoregulation, and it's how we maintain our water and ion balance. So for land animals, we lose water through urination, sweating, defecation, and breathing. Now, aquatic animals can also lose water across the body surface, and we're going to talk about that later when we have a different concentration of ions. Now, excretion is the removal of metabolic wastes. And believe it or not, those two systems, osmoregulation and excretion, are linked together. So let's focus on nitrogenous waste because we've talked about water movement before, and osmosis is pretty much the way that happens. So proteins are broken down into amino acids, we know that, and then they enter the blood, and the amino acids can be made into proteins through protein synthesis, or they can be converted to carbohydrates or fatty acids. Now, when you do the latter, when you convert the amino acids to carbohydrates or fatty acids, you remove the amine group, and it's removed in the form of ammonia. And ammonia is very toxic, and it needs to be removed. So now aquatic animals get rid of the ammonia across their gills into the environment. So that's how they excrete, through their gills. Well, terrestrial animals, we convert the ammonia to a less toxic form of uric acid or urea. Now, uric acid is highly concentrated and has very little water. Reptiles and birds, uric acid. Whereas urea, we convert the ammonia and carbon dioxide together. We join them together in the liver to form urea. So in osmoregulation, we need to maintain cytoplasm composition. The transport epithelium is a layer of cells that regulate the movement of water and ions. It's just like the Casparian strip in plants. The ions have to move through the cells, the water has to move through the cells. That's your transport epithelium. Now, sometimes the transport epithelium can focus on one or the other and specialize. Some transport epithelium only let water through. Other transport epithelia allow certain minerals to go through. Others allow a whole bunch of things to go through, depending on where the transport epithelia is. And we'll talk about that, you know that, in a moment. So the osmotic environments, when we talk about osmoregulation, we know about isoosmotic, hyperosmotic, and hypoosmotic. So we got to tie everything together. So we're going to look at the marine environment. Now, seawater is hypertonic. It has more solute, so ions either want to move into the cells or water wants to move out of the cells, right? So that's because there's a lot of salt in salt water. So the superclass agnathans, they were isoosmotic, so they had an equal concentration of salt in and out of the cells. So that means they had an equal concentration of water in and out of the cells. So the water potential was equivalent, so the water moved in and out equally, no problems. Class chondrichthyes pump salt out. The ions want to get into the cells. They pump the extra salt out of their bodies through the rectal gland. Now, they also keep urea in muscle tissues to increase the solute concentration, to have water move into the cells, and to decrease the movement of ions into the cells. There is a compound called trimethylamine oxide, or TMAO, which protects the muscles from the urine. Now, as a result, sharks are slightly hyperosmotic, so they take in water and they urinate frequently. Now, class Osteichthyes, their gills remove the salt, The ammonia exits the gills by diffusions and into the environment. So the gills are pretty much the main excretory organ in fish. In fresh water, I hope you can understand that the organisms are hyperosmotic. So the water wants to constantly get into the cells, and the ions that are in the cells want to move out. Now, paramecium, they have a contractile vacuole, and they're the first organisms to have something that helps maintain homeostasis when it comes to water. Fish are always excreting large amounts of water. This and they also lose a lot of solute. So how do they get solute back into their bodies and cells? They eat. So they're eating for solutes, they're eating for nutrition, and they're constantly getting rid of water all the time. So they're constantly urinating. They're amphibians. The larvae, they live in aquatic environments, and tadpoles excrete ammonia, but the adults excrete urea. Kind of weird how that works. So things change. Now, if we're going to talk about comparative excretion, we need to first talk about platyhelminthes, flatworms, because they were the first ones to have a specialized structure to get rid of ammonia, and those are flame cells. They are tufts of cilia at the end of tubes, and they move the ammonia out of the tubes and into the environment. Annelids, they have metanephridia, which are tubes that take the nitrogen waste out of the blood and drains it to the environment through nephritiopores. Now, they have two nephridia or metanephridia per segment. Arthropods, which are in the class Insecta, they have Malpighian tubules that remove nitrogen waste from the hemolymph, that's their blood, and place it in the digestive tract. So when they go to the bathroom, they get rid of the nitrogenous waste. In the class Crustacea, they have green glands, and they're basically the same thing as Malpighian tubules. Now we're going to talk about terrestrial environments. The main problem when moving on land is desiccation, the drying out. You need to conserve water. Now these organisms have transport epithelia. Annelids have metanephridia, right? And they're specifically geared for taking out and removing nitrogenous waste from the blood. Arthropods have a waxy layer to decrease desiccation, and they also, if you look in the class Insecta, they have Malpighian tubules, which also remove nitrogenous waste from the hemolymph and the green glands. The kidney, the vertebrate kidney, is designed to remove waste and conserve water. Now, blood is cycled through the kidney, right? And a great example of a kidney is the individual kidney or the human kidney. If you take a look at the human kidney, it lies between the 12th thoracic and 3rd lumbar vertebrae. It's about 10 centimeters or 4 inches long and 5.5 centimeters or 2.2 inches wide. And they have a little hat that goes on top of the kidney, which is called the adrenal gland. The renal artery brings blood to the kidney. The renal vein carries blood away from the kidney, and we have ureters that carry the waste from the kidney to the bladder by peristalsis. Here's a picture. We have the kidney here. On top is the adrenal gland, right? We have the renal vein, the renal artery, the ureters that come here. This is the bladder. It contracts behind the urine. We'll talk about that in class if you want. And the urethra, which carries the waste out of the body through a tube. If you want to draw that simple picture, that would be an awesome thing to do. Hit pause. The urinary bladder is the temporary storage of urine. It has stretch receptors, which tells your body, hey, I'm full, I need to pee. Now, it's full of muscles, and it contains about 200 milliliters of urine. The urethra carries the urine from the bladder out of the body. Males, their urethra is anywhere from 18 to 20 centimeters long, where for individuals, their urethra is anywhere from 3 to 5 centimeters long. This is part of the reason why individuals have problems with UTI or urinary tract infections. The bacteria have a shorter way to travel until it gets to the bladder. We can talk about the causes of UTI in class if you'd like. Then there's the external urethral sphincter. Now, a sphincter is a circle of muscles. The external urethral sphincter is a controlled sphincter, thank God, because if not, then we'd be peeing everywhere. So the blood entering the kidney is under a high pressure, and the pressure will affect something known as the glomerular filtration rate, or GFR. So basically, the blood is going to be forced, or the fluid in the blood is going to be forced out of the blood through a membrane into the kidney. Now, there are three regions of the kidney. There's the cortex, which is the outer part, the medulla, which is the inner part, and pyramids that are found within the medulla. The cortex contains the nephrons, and the loop of Henle will move from the cortex to the medulla back into the cortex. And the collecting tube ducts join in the medulla to form the pyramids. And all of that hopefully will make sense in a moment. Now we have pictures of the kidney. The cortex is on the outside, the medulla is here, and these structures are the pyramids found within the medulla. And there's the ureter. If you want to draw that picture, that would be good too. Hit pause. The nephron is the basic filtering unit of the kidney, and there are 1 million nephrons per kidney. The nephrons, or the kidney, will fill about 1,100 to 2,000 liters of blood flow through the kidneys every day, and 180 liters of filtrate are forced into your kidney every day. Now, you only excrete 1.5 liters per day, or you should excrete 1.5 liters per day. That's almost enough to fill a 2-liter bottle. So that means that you reabsorb 178.5 liters of fluid back into your blood every day. And that's done because of your kidneys. So the basic nephron, let's talk about the basic nephron here. So the nephron is in green here. This is the Bowman's capsule. It's a little like a C-shaped thing, or backward C. Then they have this little tube, which is called the proximal convoluted tubule here. Draw this, please. Hit pause and draw this. As it comes down, we're going to have the loop of Henle. This is the descending loop of Henle. Why? Because it's going down. And if you come up here, you have the ascending loop of Henle here. The distal convoluted tubule is here, and then the collecting ducts are here. Meanwhile, there are blood vessels. The blood vessels that surround the loop of Henle are called the vasa recta. Love that name, vasa recta. And then, there will be blood vessels that surround the convoluted tubules. Those are called the peritubulary capillaries. This one, peritubular, is one word. So peritubular, P-E-R-I, tubular, capillary. And they surround the proximal and distal convoluted tubules. So the general path that the fluid takes through the nephron is as follows. So the afferent arteriole will branch into a capillary that forms a structure called the glomerulus. Now, the glomerulus is surrounded by the Bowman's capsule, which you just drew. The podocytes that line the Bowman's capsule will select what enters and exits the nephron. That is your, what's it called? Transport epithelia, right? The filtrate will enter the proximal convoluted tubule, which is surrounded by the peritubular capillary, right there. The filtrate will then move down to the descending loop of Henle, which moves into the medulla, and the ascending loop of Henle, which moves back to the cortex. The loop of Henle is surrounded by a capillary called the vasa recta. And I'm running out of time, so we're going to have to end it right there. Have a good night.